Hi there. In this video, we'll talk about online informational and computational resources for biologists. We'll have a slightly different format. I'll record going to the websites and doing the searches live as opposed to having screen captures in a PowerPoint. Here are the main topics we'll be discussing today and the main websites you'll be required to know about. We'll start off talking about Entrez Gene, which is the NIH website which you should go to first for your entry point into getting information about a specific gene. We'll also talk about the European equivalent to Entrez Gene, which is UniProt. We'll talk about PubMed, which is one of the two best places to go for accessing the biomedical literature. We'll talk about exploring the genome at the University of California Santa Cruz Genome Browser. You already know about using BLAST to identify sequences, either nucleotide or protein. We'll talk about how gene function has been codified using the gene ontology hierarchy, and then we'll look at genes and proteins in their functional context in pathways and protein complexes. So we'll begin by going to Entrez Gene. And we'll do a search for TP53, which is the gene symbol for P53, the protein you know about. Here it's the top hit. It's the version from Homo sapiens, human, so we'll click on that. And here is a long web page of which we'll have to scroll down many times to see all the information. But here at the top there's some summary information. One of the most useful things about any gene is the summary paragraph that's included at the beginning. Here you can see some things, you can read it quickly to get a good idea about what a gene does. And as you can see here, it talks about how P53 can induce cell cycle arrest or apoptosis, things you're already familiar about with this gene. Another important part right here is the also known as section, because many genes go by many different names, and sometimes it can be quite challenging to know if two people are talking about the same gene or not. On the right here, there are links to some of specific types of information that's below here on the web page, but we will, rather than clicking on them, we'll scroll down and talk about the information as we see it. I won't talk about everything, which would take far too long, but also sometimes there are other resources that are better for particular types of information. One of the things that's very useful is the bibliography about this gene. It's a link to the literature about the gene P53. There's a link here that you can click to get all 6,000 plus articles that have been written about this gene. And then below that, the links to specific papers are excerpts or sentences that are taken from those papers, which can summarize quickly a result or a fact about gene function for that protein. The next thing that I'd like to highlight is here they have an interaction page and a table with genes that P53 is known to interact with. You can click on links to go directly to the page, or you can click on these PubMed links, which will take you to the paper that describes, for instance, the interaction between the protein BLM and P53. At a later moment, we'll come back to various pathways that P53 is involved in. Likewise, we will come back to the gene ontology description of the function of the P53 protein. Down below here we have another section which has other names of the protein, and then we also have the links to the definitive sequences of this gene. The NM sequences correspond to the RNA sequence of that gene, and it'll be down here. If you want to know what the sequence is, here you can see about 2,500 nucleotides that correspond to the cDNA, the DNA copy of the mRNA that is expressed. You can also click on these NP numbers, which is a link to the definitive protein sequence, shown here again at the bottom of the page. And you can see that it's about 390 amino acids. Because the gene can be alternatively spliced, there are other isoforms here, but much of the information will be very similar, and usually the top or first variant will be the either canonical or most complete sequence present. And that's a very quick intro to Entrez Gene. Thank you for listening.